and welcome to my talk. My name is Nikolaus Fortelny and I'm a group leader at the University of Salzburg. I will today be talking about reliable interpretability of deep learning and on biological networks. And with this, I mean the field of biology-inspired deep learning. This is exemplified here by the PNet model. The PNet model, but also other biology-inspired models, use deep learning where the architecture of the computational graph is based on prior biological knowledge. So here, the architecture, so the hidden nodes and the connections between them is based on prior knowledge with a sparse connectivity. So the nodes are sparsely connected. In this case, there are genes in this level, then there are different pathways in biological processes, and some genes are connected to certain pathways, but not others, and so on. In PNet, this is based on the reactome data, and then this network is used to predict from mutation data clinical outcomes, specifically metastasis. Because of this sparse connectivity, we can use node importance scores to understand which of those hidden nodes or biological pathways are relevant. And so this kind of model produces interpretations such as this one, the importance of individual pathways in the model. This is interesting because these pathways and so on are not themselves directly measured, but provide interesting hypotheses for follow-up research. Now, in this project, we wanted to test two different things of these models. We had previously established in a publication on so-called knowledge-primed neural networks, and that similar networks are often not robust when iteratively trained, and also are affected by network biases. So in this project, we first tested the robustness of the PNet model. And robustness here means that if I train the same network on the same data multiple times, whether I get the same results. So here is one network, and before training, we have randomly initiated weights, and after training, we have trained weights, and we get the importance scores of the nodes. But when we train multiple networks, they get different initial weights, and then they get different importance scores. Of course, you can also set the seed to have the same initial weights, but this and this and this starting network are equally relevant. So it is relevant to see whether those three different networks provide the same or different importance scores. So then we get those different importance scores at the end, and we compare them and see whether they're robust or not. And you can see this at my poster. The second thing we wanted to test is network biases. So certain networks have more, certain nodes in networks have more or less connections than others. In biological networks, that's very well known with hubs and so on. And so we developed two control approaches to control for network biases. In one approach, on the left, we use deterministic inputs. So we create artificial inputs where every input is perfectly correlated with the label. So all the mutations, all the CNVs are perfectly correlated with the label. So the network doesn't have actually relevant data, it just predicts based on network biases. In the second approach, we randomly shuffle the label, so there is no predictive power. And then again, the network is not able to train on data, but just based on network biases. And we compared these effects on interpretations as well. Details, again, you can see at my poster. The conclusion of this work is that initial weights do affect interpretations, so we need to assess that to limit robustness. Network properties affect interpretations, so we need to control for network biases. We found both in KPNNs and in the PNet model, and we think they likely affect all biology-inspired models, but they're often or almost always ignored. And the take-home message is to repeatedly train your algorithms and think about the right controls. Now, with this, I would like to close, and I would like to thank my group at the University of Salzburg. Here are some pictures from our most recent lab hike, and I would specifically like to point out this person here, Wolfgang Esser-Scala, who has been performing most of the analysis presented here today. We are currently in the process of publishing this article, so you may see it on bioRxiv by the time you watch this video. With this, thank you.